Okay, great. So we have 4 panelists up here. We have Savas Tay, we have Peter Nemes, Ryan Kelly, and Henry Brinkeroff. So we have plenty of questions that we have prepared, but perhaps someone wants to start off the session. But I can ask the first question of how can we make single cell proteomics more accessible for people to practice, right? We're a small community still, I think, and we can make it even larger. So what are your thoughts here, if anyone wants to start?
So I have a perspective on this because of my current work, which I will talk about today in the afternoon. But I think one of the main challenges is the difficulty in the technical approaches that we take. You need specialty equipment, specialty tools, which are sometimes a little bit difficult to adopt. So simplifying the readout platform, maybe even like when you start designing your technology, thinking a little forward and try to adopt a simple readout modality from the get-go might really enable a widespread use of single-cell proteomics and ultimately democratizing the technology. So that's where I stand.
Thank you. I'll just add additional points. I think automation is number one. And perhaps opening single-cell proteomics course facilities would be another way to go. In many times, these technologies are developed in unique laboratories. Up until we get fully automated systems, I'm pretty sure Ryan will comment about that. That's one way around. And we oftentimes get a lot of requests for collaborations where samples are still quite different from each other, different types of cells, different types of cell sizes, different types of specimens. And so having a facility, for example, where dedicated personnel can figure out those technical details routinely would really open up, I think, the throughput and bandwidth for bringing proteomics to a broader community. Pretty much. Ryan, I'll pass the mic now to you.
We'll just go down the line. Is that good?
Yeah.
So, well, I just thinking about LC, but right now anybody can do sample prep, anybody can do LC, and anybody can do mass spec. It's just there are trade-offs between what's easy right now and what is, pushing the absolute limits. But these things move in a trend, right? When I started in mass spectrometry, the default separations this was a cutting edge, Dick Smith's proteomics lab. It was 2 microliters a minute with 150 micron columns, and I was pushing it, and then people figure out, oh yeah, actually, this gets easier over time. We just figure out ways to do it easier. And to be a mass spectrometrist 40 years ago, you were building the whole instrument, right? And so in many cases, there were commercial options, but being a mass spectrometrist meant you did all the electronics and hardware yourself, right? And so just over time, what's really bleeding edge right now will become routine. It's just the way things go. We figure out easier ways to do things. And I think, we'll see that with sample prep, separations, and our mass spec vendors are making things better all the time too. So it's just, I guess, patience. I don't know.
Yeah, I would add, I think that the simplicity and streamlining is definitely a democratizing force, but also I think just bringing costs down, because there's a big difference between, oh, I can do this, at a reasonable time scale and a reasonable expense if I have access to like a major facility with many $1 million mass spec machines, or complicated multiplex systems. But there's a huge difference between that and, this is something that you can do in a laboratory, as a starting PI at an institution that doesn't have access to those kinds of resources. So I think also just new technologies that, can reduce costs for specific biological applications.
Does anybody in the audience would like to comment? See.
Follow-up question. Build up that. Yeah, so kind of a follow-up question to build off that a little bit. And if, it seemed to be that access to the equipment and, expertise for doing various steps in the workflow is important. And I guess given that we're in Boston, there's kind of this large biotech presence. Do you see a potential for, outside of companies who are manufacturing the instruments, do you see potentials for a company to play a similar role where they kind of facilitate maybe if not end to end, a large portion of the workflow to kind of disseminate the technology more widely and fill in gaps where, institutions that might not have a mass spectrometry core that's able to provide all of these can kind of fill gaps for people interested in that way.
I mean, I think that's something that's definitely happened in genomics, at least. I mean, coming from my background in that field, oligosynthesis is something now, you can, 50 years ago, very hard to do, but nowadays, I can put it in order, costs $50 or something, and I can have 100 nucleotide oligo in next day. And a lot of that, that's not done because we all have oligosynthesis machines in every lab. It's done because there's, I think a lot of industry support for these kinds of things, and these centralized facilities in a commercial context, I think it can definitely be really useful. Can't speak too much to mass spec, but just in general, I mean, once you have, I think, a defined set of common, uses of tools or biological questions, that's when you kind of think start streamlining things like that.
I can add a little bit to that. Yes, I think ultimately it's going to be the industry commercialization that will enable these things to be really adopted by a lot. On the previous point, all the talks that I heard today and yesterday points to a low-hanging fruit when it comes to improving single-cell proteomics. I think that's the sample prep. There's a lot that can be done on the sample prep end. And there are all these, kind of orthogonal fields from microfluidics, for example, that have been developing and developed a lot of capabilities. So if we can find a way to merge these, single cell mass spec with microfluidics, I think that can be major leaps forward in democratizing this.
Of course, none of these developments would be possible without a good amount of cash infusion from funding agencies, right? I mean, that was one of the reasons why genomics took off so well, because they got the support of the community. So if you're a peer reviewer, if you're in a funding agency, that is something that our field will desperately need to be able to bring these innovations to access to pretty much everyone. This is an expensive field, not just because the instruments are expensive, but because the required expertise takes a long time to develop. A student takes 5-6 years to graduate with the amount of expertise needed to do proteomics in terms of sample preparation. Another student, for example, for bioinformatics, another student potentially for mass spectrometry. So if you really want to make this technology as a push of a button type of approach, that will take substantial developmental efforts. And I think we can get there, provided we've got your help, your backing.
So I maybe have a question actually on this topic.
Go ahead. What Peter said is a perfect segue for another question that they wanted to ask, but go ahead, Henry, and then I'll ask.
So as a kind of novice to mass spectrometry, I'm curious, as far, is it common, so with genome sequencing, you usually have there's a kind of a generalized question you can ask, which is, I'm going to send you a vial of nucleic acids, what's the sequence of the nucleic acids, right? But I feel like the hunch I've gotten from hearing all of the wonderful talks is that mass spectrometry is quite a bit more complicated, and it's sort of an exploratory process a lot of the time where you set out, you start measuring something, and then your initial measurements inform how you proceed to analyze the data and think about it and what to measure next. And so I'm curious, do you think the bottleneck to some sort of commercial implementation is really like a technological hurdle, or is it more of that, most of the experiments people are currently doing are so variegated and so different in their objectives that it's hard to have some sort of unified system for doing it? That's, I guess, open question.
Yeah, that's kind of the blessing and the curse of mass spectrometry, is that it can be applied to such a diversity. The same tools you can do, chemical cross-linking and all the, ask so many questions, but there's not such a defined one goal that everybody's after that would streamline and unify things. So yeah, kind of goes both ways, I think.
We have a question from the audience.
Hi, this is a little bit of a follow-up and then a second question. So the experts here in this room, have you thought about putting together a workshop? Several years ago, when I was looking around how to learn metabolomics, a Cold Spring Harbor workshop was the thing that really changed my life and pushed me forward, and saying you need experts. You need to disseminate that information out there for everyone who is willing to take it. So that's one thing. And the second thing, sort of different topic, but I want to throw it out there, is what do you think are the killer applications that would show the power of the techniques that we are seeing in these 2 days? That would sort of drive the motivation for funding, that would drive the motivation for developing the technique further.
In the first point I'll comment on, I'll leave my colleagues to comment on the second. Outreach is really important, I think, in our field, since many of us come from analytical, bioanalytical technology backgrounds. The Cold Spring Harbor has a nice single cell course, and I actually have participated in that too. It's really exciting to see when you meet with biologists, especially, and translational scientists, how they think about the problems, especially pertaining to single cell proteomics, single cell metabolomics, and single cell peptidomics too, that we haven't really talked about today. We have a lot to offer, but only if we bring ourselves to the other communities will they recognize the potential of the tools that we've got. And so outreach is really important. The second point is very important. How do we make this technology amenable to broad types of applications, essentially? And we work with different biological model organisms. I work with frogs, mice, and so on. But there's many more types of single cells, T cells you've heard about. They're all going to come in different forms, in different shapes, in different substrates, in different media. And so being able to demonstrate all of these applications would be cool. Perhaps maybe a community-wide paper, a white paper would be helpful to that end. And also creating guidelines to the best ways of doing single cell proteomics, single cell omic measurements, what are the minimal requirements to publish for reproducibility. Nikolai actually is leading an effort to that extent, so we hope to be able to report on that sometime soon. If there are any other feedbacks, I'll pass the microphone.
Regarding the second question, I believe there will be clinical applications of single cell analysis at one point. But currently and in the near future, I think the biggest applications will be on fundamental biological discovery and also in computational modeling and prediction of biological systems. All of that data that we are generating in this area can really be used to come up with predictive, accurate models of complex biological systems and used for discovery. That I think is the lowest hanging fruit when it comes to single cell analysis applications.
Yeah, Frank Larkin from Bruker, just because you talked about mass spectrometry, so I thought I'd give you a little bit of a perspective. I don't see any trend yet for this all to get distilled into one method, right? We started out with scopes and other forms of labeling, unbiased single cell proteomics. There's now other diaplex versions, new methods that are coming along. People do untargeted. For those unbiased single cell proteomics, which we're mostly by implication discussing here, mass spec sensitivity, dynamic range are very important. What's happening at the front end, sample prep, single cell isolation, whether it's by laser capture or it's already single cells in blood or something like that, tremendously important, and a lot more progress and automation needed there. At the back end, new methods, like on our platform, at least the dia-passive method with a new processing method, whether it's Spectronaut or something like DINN. Many of you, the experts, are familiar with all of these things that are key contributors to higher sensitivity, to higher throughput, to higher performance. And then, as described earlier by Theodora, there's now unbiased spatial single cell metabolomics or lipidomics. That's a totally different instrument, right? That's a 5 micron resolution MALDI instrument. It may have a timsTOF analyzer, but it's a different front end. Similarly, there are now brand new targeted proteomics methods that also use that 5 micron resolution. These Ambergen panels that we now use for high-plex IHC for looking at 40, 50, maybe in the future, 100 targeted proteins with single cell resolution. So if anything, the field is diverging and getting broader. And so far, the trend is for more capabilities and more flexibilities for the unbiased versus targeted proteomics versus unbiased metabolomics approaches. That's my observation for now. In 5 years, the trends may be different, but for now, I think people are looking, we're at the very early, getting into the very steep part of the development curve. At least we're still in there.
All right, Nikolai Slavov also has a question or comment.
Yeah, I have a quick comment in response to Frank, and also have a question in response to what Peter said. So my comment for Frank is I very much agree about fundamental differences between imaging methods and some of the electrospray methods being performed on different systems, and they're different enough so that one needs specialized equipment, specialized knowledge. But also there is enough similarity between many of the single cell proteomics methods, such as label-free, PLEX-DA, scope, prioritized scope. They can be implemented on the same equipment in the same lab with relatively minor changes in how we program the instruments and do the data analysis, at least from my perspective. And I tend to be a bit of a unifier. I tend to try to see how we can simplify things and also convey the message that some of that high degree of specialization and divergence is apparent, but fundamentally there is also the opportunity to unify it. And of course, there are the big differences in hardware that between imaging methods, electrospray methods, that are more fundamental and require, yeah.
On the same platform, but
yeah.
It's different configurations on the same
platform,
yeah. Not totally different features.
And I think another aspect in the discussion of how do we make the methods more accessible is also educational, really having a way to convey to the broader community the core principles, the big ideas, so that even non-experts can know what are the capabilities that they can use in their experiments and to more effectively interface, collaborate with experts. So my question for you, Peter, that I wanted to ask, speaking of funders, is I happen to know that there are some funders listening to our conference, and I'm interested to hear your thoughts on what advice you have for them. What are your thoughts about directions for research that funders should support? And what's your advice for them?
Think boldly and believe in us. We have come a really long way just in a decade, basically, maybe 2 decades in mass spectrometry, especially in single cell analysis, thanks to developments in sensitivity, specialized workflows. We are really on the right path, and I think we can make this work. But it does take a team effort, not just community-wise, but even within one laboratory. You just saw, for example, slides earlier from bioinformatics to mass spectrometry to sample preparation. It's really a one streamlined approach we need, and for that we need support to be able to sponsor different developmental trajectories. This is not just about buying a mass spectrometer, but sustaining an entire research group financially, right? And then promoting interactions within the community. So buying instruments is important, supporting personnel is equally as important, and then finding or creating programs where experts, leaders in the field of mass spectrometry can join forces toward disseminating our tools and technologies and methods. And that's, I think, really what's going to allow us to essentially catch up to where single-cell transcriptomics stands at present, in large part because they got the support already over the last couple of decades. So thanks for listening, and we look forward to your funding.
I love the question on what's the killer application too, and I think there could be killer applications across the NIH space, but tumor heterogeneity is one of those, and NCI has been pretty proactive in funding single cell proteomics research with new initiatives. They have with NHGRI this joint single cell proteomics and single molecule protein analysis call out there. So I think the funders get to kind of dictate where the killer application is going to come from to some extent. But I think as a community, we would really, really benefit from that killer application. Probably won't be coming out of my brain, but I do see tumor heterogeneity as kind of an obvious, kind of otherwise intractable problem to go after. So.
Okay, cool. We actually have a question on Zoom from Theodore who just spoke. So if you want to unmute yourself, you can.
Yep. So, yeah, I would like to chip in, but also kind of like reinforce this discussion about killer application, which was actually very interesting to follow. So, and heterogeneity is great, of course, for single cell. But I think what's even more interesting is post-transcriptional effects, right? Because this is probably what will be really not only kill application, but the thing which is possible to do with either proteomics or metabolomics. And for metabolomics, we're definitely looking into non-genetic factors like, say, drug uptake, right? So which is very applied, but this is where we can detect drug metabolites and so on. And also effect of like pesticides, anything else which is detectable by mass spec. So my question, a little bit to you and to the whole audience. So what is now your, the best results, about the consistency, but even in particular inconsistency between proteomics and in particular PTMs and proteoforms readouts to transcriptional programs, right? Because this is what is killer for this community, everything which comes beyond the transcription per se, right? So, and do you see how much of this also you see on the single cell level, right? Because in particular, we're talking about PTMs, which is would be the brilliant, right? And at the same time, it's so dynamic as well. So, so how much you see also single cell proteomics evolving in this direction, providing these readouts which are not possible at all with a single cell transcriptomics?
The sort of model system that I've thought a lot about in developing, single molecule proteomics approaches and just talking in that community is the one I'm most excited about is MHC peptides. These short peptides that signal on the outside of a cell to the immune system, recognize, for your own body's immune system to recognize, whether to destroy the cell, whether it's healthy or cancerous or infected. But yeah, these short peptides, they are often spliced, it is thought, by the proteasome into all kinds of different weird configurations. They have heavy PTM decorations that actually are functionally super important, and none of this is in the RNA transcript, in the transcriptome at all. So I think it's a big question that proteomics is really the only access point to. It has huge applications in like cancer immunotherapy, vaccine development, all kinds of stuff, and just fundamental understanding of immunobiology.
Another question from the audience.
More of a comment than a question. So the question before was about the role of PTMs and how they are important, or protein-protein interactions would be another application. And I just want to say that even if it's not going to be possible soon at the single cell level, you guys should take the credit for improving the technology, that even if we end up with using 100 cells, right, with PTM enrichment and for protein-protein interaction profiling, that will be possible because of the technologies that you're developing for single cell, right? So a lot of these things will translate to 100 cells or even 1,000 cells, right? So in that sense, I think, all of this that's presented here, in my view, is equally important, even if it's not going to be just single cell analysis.
That's a great point.
Thanks, Alexi. And also, so we're not just in single cell mass spectrum generating large amounts of data. We're generating useful information that can inform functional biological experiments, but also can inform the development of next generation probes. Right. And Alexi raised this point that if we are not able, for example, to measure protein-protein interactions at a single cell level just yet, but we can do that at the level of, 100 cells, that can help the development of specialized probes, antibodies, other kinds of probes that then will allow to go into at a single cell level. So I think our technologies in terms of mass spectrometry can really drive future technological developments outside mass spectrometry. And it's a really exciting integration of multiple orthogonal developmental trajectories.
So I also have a quick comment in response to Theodore's very insightful question, with whose premise I agree. I think that there is clearly the possibility of extending our measurements to various forms of protein modification, whether that's by covalent attachment of phosphate, acetylation, and so on, or just proteolysis. And today we are going to see examples of the technology already achieving that. But even beyond, before we measure comprehensively, deeply protein modifications, there is also the possibility of analyzing regulation at the level of protein degradation, protein synthesis, simply by comparing in a meaningful way measured RNA and protein abundances. And Alex may want to comment on that, the extent to which that's feasible. You want to briefly talk about it?
About protein degradation, or?
About inferring post-transcriptional regulation from joint RNA and protein measurements.
I mean, it's really exciting application to be able to either take single cells and split them in half and measure the RNA and the protein, or basically you can do sort of a more paired analysis where you have single cells from the same population undergoing single cell RNA sequencing or single cell proteomics. And I think there's a lot of different ways to think about this, right? Because if you can get both types of information, we can talk about dynamics and basically see how we've talked a lot about with Andrew in the back here. We're talking about how we can sort of track the rates of unspliced and splicing of mRNA, but then also adding that information with the proteome. So if you can get those two rates together, that'd be interesting to see if there's any protein sets that have maybe slower, transcription, but faster translation, or vice versa. Yeah, go ahead.
Yeah, so a lot of interesting biology really happens through structure. So it's difficult to ignore structure, protein structure. So, and although I know a lot of people in this audience do not like to work with things that work through structure, like antibodies, they do exist and they can give you a lot of information on what is binding to what, complexes, and also open the way to doing joint measurements of transcriptome and the proteome. So I think these techniques that on one hand really measure proteins directly, and on the other hand through binding partners, would be complementary with each other and then generate increased insight into post-translational aspects of biology.
Yeah, for sure. Does anyone have one last question? I'm not sure if we're running out of time, but one more question.
I was interested in further comments about what Peter called automation, right? So do people think that the future is more around like a single turnkey platform that provides samples to answers? Maybe Brooker provides a more complicated microfluidic front end to their system? Or is it more of each subsystem is optimized individually, and then there's some centralized robot or some other method of transferring samples between the various subsystems?
All of the above. I mean, automation would definitely help us in terms of sample preparation. I don't know about a single platform that's going to go from start to finish. That takes a lot of effort, even in transcriptomics, right? You still are handling things many times manually before you use a sequencer. I mean, many of us are still doing single cell isolation sampling manually. So if there are ways to automate it, that would be a huge first step. While adherent, so cells suspended are a whole different topic to analyze than tissue-bound single cells. Do we now envision a single platform to deal with all kinds of samples? I don't think so, right? If you have a cell suspension, you might be thinking about fluorescent activated cell sorting, type of instrumentation for high throughput introduction and processing, maybe via microfluidics combined. But if you're working in an actual tissue environment, and if you want to preserve spatial and temporal coordinates, and that was one of the message I hope I delivered, then you have to think in a different way. Right now you have to register coordinates in space and time. And that's not easy, but it also could be automated, right? I mean, we saw imaging mass spectrometry can provide you with that information. In fact, Jonathan Sweetler is another exciting researcher in the field who has also used imaging-based MALDI, right, where the coordinates of cells could be recognized, and then MALDI could be governed for even higher throughput. So there are so many different ways we can tackle these challenges. I'm also not sure that a single technology is in everyone's interest. The more technologies we have, the more opportunities we actually have for progress forward, and also the better kind of security have that if one technology fails in particular niches of applications, the other one can step in. So putting all the eggs in one basket is not always the best strategy, as we know.
It is kind of exciting to see these, not one company providing the whole workflow, but at least these companies forming kind of alliances from, maybe EvoSeb with Ion Optics, with Bruker, with, and Selenium at the front end to make sure that there's at least a path forward to make these measurements with only commercial instrumentation. It's not like you have to use that, but it's nice that they're playing nice together to provide this kind of end-to-end workflow. I'm not currently using that particular one, but I think that's going to drive accessibility and dissemination of the workflow quite a bit. So.
Well, that's really quite awesome. We had a really great discussion. If we have any more questions, we can bring up to our coffee break, but let's thank our panelists for such a great discussion.